Hey all, this is the second video in our series on biotechnology. In the first video, we talked about how to create a cloning plasmid, or how to put a gene from, for example, human cells into a plasmid that we would eventually express in bacterial cells. In this video, we're going to talk about how we actually get that plasmid into bacterial cells, and then we'll talk about how do we determine that we've actually successfully cloned the gene that we want to. So we'll talk a little bit about sequencing techniques. So let's talk briefly about the overview of this process. First, we're going to take bacterial cells that are competent and we're going to mix them with the plasmid and we're going to transform them through the process of transformation. This is going to allow for the plasmid to be taken in by that bacterial host cell. Next, we would isolate the plasmid from these bacterial cells, and since we're growing these bacterial cells, we can isolate these plasmids at high concentrations. And we can take that plasmid then, and we can do a couple of things with it to confirm that it does have the gene we want in this cloning plasmid. So first, we would probably end up doing a restriction enzyme digest, and then taking the fragments of that plasmid that have been generated from the digestion and running them out on a DNA gel electrophoresis. And we'll talk about this process in detail. Typically, a follow-up to this then would be a sequencing reaction, where we would design primers specific for the gene that we have placed into the cloning plasmid, and now we would determine, does it have the correct order of A, G's, T's, and C's? In other words, is it capable of forming a functional mRNA and a functional protein? E. coli is not a naturally competent species of bacterium. Instead, to make them competent, we have to chemically treat them. Chemically treated cells that are going to be transformed by heat shock are known as chemically competent cells. E. coli that are chemically competent are incubated with the plasmid on ice for a short period of time. Afterward, the temperature is increased to heat shock these cells, and that heat shock is going to open up the pores in the bacterial membrane to allow for DNA to diffuse into the cell. These cells then are rapidly chilled back on ice to close up those pores and to basically lock the plasmid inside the cell. Now, we can also transform E. coli and some other bacteria by another method, which is known as electroporation. In this case, we generate electrocompetent cells, which are treated with reagents like glycerol. We incubate them on ice with the plasmid, and then instead of a heat shock, we use a small dose of electricity through a process known as electroporation. The electricity here, in the same way as the heat shock did, is going to open up those bacterial cell pores to allow for DNA to diffuse into the cell. After electroporation, freezing these cells or cooling them down on ice is going to help to close up those pores to allow for those bacterial cells to recover. The next step in the process then is to isolate or to enrich for those bacterial cells that have been transformed. What you'll notice is on the left we have a population of cells, some of which contain the plasmid and some do not. You may also recall that in the past we've talked about how these expression plasmids contain on them genes that render bacteria resistant to antibiotics, or that can be selected in some other way. And so if we take that population on the left there, after recovery, we can go ahead and select them using antibiotics. So for example, the cloning plasmid that we talked about contained the beta-lactamase gene, which encodes for ampicillin resistance. In this case then, we can select for the bacteria that contain the plasmid. If we grow bacteria, for example, after transformation on LB agar with ampicillin added to the media, what we would find is that each of these colonies that we have here, including these three that I'm highlighting, are ones that have likely been transformed with the plasmid. However, we do not yet know whether that plasmid does contain the gene that we want it to contain, for example, the insulin gene, going back to our last lecture, or whether the sequence of that gene is even correct. And that's where we need to follow up with additional experiments to confirm that these transformed bacterial cells are indeed going to express the gene that we want them to express. Here is the plasmid that we set up to design. Through the cloning process, we wanted to place a gene downstream of a transcriptional start site with a promoter. And we also had in here the beta-lactamase gene, shown in orange, that would render bacteria resistant to the antibiotic ampicillin. But there are other potential combinations that could have been generated because of mistakes in the cloning process. For example, we could have cloned the plasmids that are shown here. In the bottom at the middle is a plasmid that does not contain our gene of interest. And so maybe it's possible that this plasmid religated to itself without including our gene of interest. It's also plausible, though unlikely given our cloning strategy, that the gene we tried to clone into this plasmid would have been placed in flip-flopped from the direction that we wanted it to be. And so how do we distinguish between these three different types of results? So let's imagine in this scenario that we're going to use the restriction enzyme BAMH1. In our original plasmid, which again is shown at the bottom here, let's imagine that there are two BAMH1 sites. If this plasmid were 4.5 kilobases in size or 4,500 base pairs, then maybe this would produce two fragments of 2 kb and 2.5 kb when the plasmid is digested with BAMH1. Let's now take a look at the scenario which we're hoping to achieve, in which we've put the gene in the correct orientation downstream of the promoter. This again is our plasmid in the top left-hand corner. In this example here, let's assume that the gene is about 500 base pairs, increasing the size of our plasmid to 5 kb or 5,000 base pairs in total. And let's assume that this gene also has an additional BAMH1 site within its sequence. And so if this were the case, then we would expect that if we were to digest this plasmid with BAMH1, it would produce three fragments of 2 kb, 1.7 kb, and 1.3 kb. If this gene were put into the plasmid in the wrong orientation, as is shown on the far right, you'll see here that the location of that BAMH1 site within the gene in relationship to the two BAMH1 sites within the plasmid would change. And so in this case here, though the plasmid is still 5 kb in size, the fragments that would be generated would be different in size compared to the plasmid we anticipate to create on the far left-hand side. Now, the way that we would detect these different size fragments would be using a process that's known as gel electrophoresis. In gel electrophoresis, we create first a slab of an agarose gel, which has the consistency of about 2-day-old Jell-O. And that is what's being shown here in gray. Once this gel has solidified, we would place into the well the DNA that we have just digested with restriction enzyme, which is shown in blue. We then apply an electrical charge to the gel electrophoresis. And you'll notice the direction of this. The negative end is where the DNA is currently located, whereas the positive charge is located at the far end of the gel. And there's a reason why we orient it this way. DNA, you may recall, has a sugar phosphate backbone, and the phosphates in that backbone are negatively charged. And so if we apply this electrical current here, DNA is going to be repelled from the positive charge and is going to be attracted to the negative charge. And so DNA is going to be separated through this agarose gel. Now, the agarose gel is kind of a meshwork of gelatin, and so molecules are going to migrate or move through this gel depending on their size. Fragments will begin to move all together, but what we find is that as we allow for separation, fragments that are smaller are going to migrate through this gel faster than fragments that are large. And so in the end, we will be able to separate these fragments of DNA based on their size. The largest ones will be closest to the starting point where the well was located, and the smallest fragments will have traveled the furthest down the length of the gel. So now what we're going to take a look at is a simulation of the results that we would expect to find. On the far left-hand side, you can see we have a lane that's labeled as ladder. Oftentimes in DNA gel electrophoresis, we use a standard ladder with fragments of known sizes to compare to our samples. So for example, this is what we would call a 1 kb ladder. We have fragments of 0.5 all the way up to 4 kb. Let's assume that clone 1 here is a clone that contains only the plasmid and not the gene that we wanted to clone into that plasmid. You'll recall from our previous slide that this would produce fragments of 2 kb and 2.5 kb. And so if we were to run a gel electrophoresis, we would see products that look roughly like this. Clone 2, let's assume, is a clone that contains the expression plasmid, but the gene is in the opposite direction of what we wanted in. We said that this would produce 3 fragments. 2 of those fragments would be about 2 kilobase pairs in size, and the third one would be 1 kilobase pair in size. And I'm showing that here for clone 2. Let's assume clone 3 then is the clone that we expected to make from our experiment. We said that the gene would be coded in downstream of the promoter in a direction that would, if we were to digest this with restriction enzymes, produce 3 products, one of 2 kb, one product of 1.7 kb, and then a final one that is around 1300 base pairs. You can see by using this process that we've been able to distinguish these 3 different clones from each other. And only clone 3, shown here, contains the correct pattern of DNA bands that we would have expected to see if we successfully cloned our plasmid that we wanted to create. Now, it's possible that during the cloning process, particularly the PCR amplification of cDNA, we may have generated mutations within the sequence of the gene we were trying to clone into the expression plasmid. And so we need to double check for that now. And we're going to use what's known as Sanger sequencing, or a modification of Sanger sequencing. In this particular type of sequencing, what we use are dideoxynucleotide triphosphates and deoxynucleotide triphosphates. So let's take a look at the structure of these real quick and talk about why do we use both of them. Normally, in DNA polymerization, we use deoxynucleotide triphosphates, and so here's an example of that. And as we've talked about before, these deoxynucleotide triphosphates are defined by this hydroxyl group at the 3' carbon. This creates the location where phosphates are going to attach to this sugar. However, dideoxynucleotides lack this OH group and instead just have a hydrogen. These dideoxy or DD-NTPs are called chain-terminating nucleotides because nothing can be attached to them. And so in sequencing reactions, what we have are high amounts of deoxynucleotide triphosphates and low concentrations of dideoxy CTP, dideoxy GTP, dideoxy TTP, and dideoxy ATP. And the reason why we have low concentrations of them is because we only want them to stall or inhibit polymerization at a low frequency. Each of these dideoxynucleotides also has some sort of fluorescent marker located on them. And so what we can do is we can run a sequencing reaction, which is going to produce for us a bunch of fragments of DNA of different sizes each with a different chain-terminating dideoxynucleotide. These fragments can be separated by electrophoresis, and then they can be read using a laser, which will detect each of the different bases, because those different bases will fluoresce a different color under the laser. Now, what we typically see in a laboratory setting is that this work would be done behind the scenes. And instead, what we would get is a chromatogram like this one shown at the bottom here. In this chromatogram, what you can see there are peaks. Each of those peaks represents an intensity, or how frequently a nucleotide was detected at that location in the DNA sequence. In this particular one, you can see that green peaks associate with A's, reds associate with T's, blue associates with C, and G's associate with black. Now, there are newer methods of sequencing that are much more high throughput. These are called next-gen sequencing techniques. We're not going to discuss that in this particular lecture, but if it is something that interests you, please feel free to go ahead and look at it. All right, so let's go ahead and just take a look at this process one last time. I'm going to talk very briefly about this through the perspective of what I was doing in graduate school when cloning plasmids to express them in human cells or bacterial cells. So again, we take bacteria and then treat them with chemicals to make them competent. Those competent cells can be transformed, allowing them to take in the plasmid. We can isolate these plasmids from clones that we think have successfully been transformed. And typically what we would use is a two-step process to confirm that the plasmid is what we expect it to be. First, again, we'd use restriction enzyme digests and gel electrophoresis as a way to come up with a rough map of the plasmid, make sure that everything is in the correct orientation. And then secondly, we'd go back and sequence our gene of interest in order to make sure that there are no mistakes in the sequence and that we haven't created any mutations in the process of cloning. After this plasmid is good to go. You're ready to use it in your research projects. So with that, take care, well wishes, and I'll talk to you soon.